Okay, so today we're going to talk about genome editing, specifically with CRISPR-Cas9, because it's sort of the state of the art in terms of where genome editing is. And it has really gained a lot of attention and I think warranted enthusiasm in the past 5 or 6 years. So brief overview of how the talk's going to go. I kind of broke it into two sections, and stealing from Men in Black references. We're going to go with the old and busted way of doing it first, and then the new hotness, which is CRISPR-Cas9. Specifically, when we talk about CRISPR-Cas9, we'll break it down into its little individual components that form the CRISPR-Cas9 complex and talk about the role of each of them when we get to CRISPR-Cas9 mechanism. And then finally, as the last part of this section, we'll compare it to other similar technologies that have also emerged in the past decade or two, just to give a sense, an even better sense of why this is so exciting. And then at the very end of the lecture, I'll provide you guys with some additional resources too. All right, so let's start out with the old and busted way of doing genome editing. So genome editing is kind of a broad term, and it really is what it sounds like. It's basically anytime there is a modification to an organism's DNA that could be considered editing that organism's genome. So things like gene therapy, which you heard about last week and you're going to hear a little bit more about today, are in the purview of genome editing. But so are things like creating a knockout library of mice, where you're going systematically through every known gene in the mouse genome and having a mouse or a frozen mouse embryo with that knockout on tap for when somebody orders it online. That's also a type of genome editing. Okay, so it's kind of been the one aspect of genetics that has been doable for a very long time, but every system has these sort of unique ways of doing it that are somewhat idiosyncratic to the system. So for example, Drosophila melanogaster have the P elements, which are transposons that don't occur in other organisms, so it's really hard to make transgenic organisms using P elements if you're not using Drosophila, and so on and so forth with just every organism. So it would be nice to have a technology to edit a genome that is something that you can use in a huge array of organisms and do it with ease. So given that, I am going to focus the examples today on gene therapy applications, both because I think it's a really exciting aspect of something that CRISPR-Cas9 will make a big difference with in the coming years. But also because a lot of people in the class are sort of medically oriented, so it might be an exciting example. So why don't we travel back in time, Magic School Bus style, to the 1990s, where the first clinical trials, the clinical gene therapy trials were beginning. So there were a lot of candidate diseases for gene therapy, right? And to be a good candidate for gene therapy, the genetic disease has to be caused by a single locus, and that locus has to be known. But also the tissues that genetic disease affects should be tissues that are pretty easily accessible by the technologies that are going to be used to apply the gene therapy. So there was a particular disease called SCID, or severe combined immunodeficiency, that became a really popular candidate for gene therapy in the 90s. And severe combined immunodeficiency is caused by any number of loss of function mutations in a couple of genes. ADA is a really common gene where if you have a loss of function mutation at ADA that's homozygous, um, uh, ADA stands for adenosine deaminase. Then what happens is there's disturbed development of functional T cells and B cells. So essentially, individuals who have severe combined immunodeficiency don't make the uh, the immune cells that are involved in adaptive immunity, right? So they don't have an adaptive immune system. So a lot of individuals who have SCID succumb to opportunistic infections by the time they're about 2 years old or so. So early on during infancy, they're protected essentially through maternal immunity, right? So they get some immunity from their mom. But when they have to start producing their own T cells and B cells, that's when it becomes problematic, right? So it's essentially fatal for almost anybody who has SCID really early on in life. So they know in most cases which gene a particular individual who has SCID is the loss of function gene. And the good news is that it's a genetic disease that affects blood cells, right? It affects hematopoietic stem cells, which make immune cells, which make T cells and B cells. So that means that they have easy access to the target cell type, which are these which are found in the circulatory system. So it's really easy to deliver a vector carrying a functional copy of, let's say, the ADA gene, and just inject it into an individual's bloodstream where it will readily encounter the cell type it needs to encounter. Okay. So there was a French team that started out these clinical trials. They had 20 patients in the first trial, and they injected these patients with a viral vector. Okay, so imagine, right, that your job as a researcher is to try to figure out how to deliver a functional copy of a gene to a particular cell type. Okay, so you have to find the cell, bind to the cell, infiltrate the cell, vector the DNA into the cell's nucleus, and then insert it into the genome of the cell. Well, it turns out that there are organisms that exist that already do this, and they are retroviruses, right? So that's exactly the replication cycle of a retrovirus, right? Where it finds specific target cell types, infiltrates them, inserts RNA genomes or sometimes DNA genomes, but in the case of a retrovirus, it's an RNA genome, along with reverse transcriptase, which reverse transcribes the RNA into DNA, vectors the DNA into the cell nucleus, and then using a protein like an integrase, will integrate that DNA, that viral DNA, into the host cell's genome, and rely essentially on the host to transcribe that viral DNA to finish the replication cycle. So what researchers did is they leveraged these retroviruses and essentially rendered them non-replicative, right? So they can't replicate inside the host, but they can still deliver our desired transgene, in this case a functional copy of ADA, to a target tissue type. Okay, so the virus that this French team settled on was called Maloney-Murine leukemia virus, or, well, I don't know how to pronounce the acronym, so I'm just going to keep saying the full name, but that's what it looks like. So they modified that virus so that it couldn't replicate inside the host, but it could still bind to hematopoietic stem cells and insert the transgene, the functional copy of ADA, into those cells that it infects. Okay, so that looks like this. So here we have our Maloney murine leukemia virus cell. It has a bunch of surface proteins which bind to cell surface receptors on the target cell, which again is a hematopoietic stem cell in our case, and will insert its RNA copies of its genome into the cytosol, where it gets reverse transcribed by reverse transcriptase, because it's a retrovirus, shuttled into the nucleus and integrated into the host genome. Okay, so now this cell has, and all of its descendants will have a functional copy of ADA. Okay, so there were a lot of obstacles in those early studies, which included the fact that the Maloney murine leukemia virus did a really good job in vitro editing the genome of these hematopoietic stem cells, but was not nearly as efficient at doing it in vivo when they actually injected it into live patients. Okay, and their choice of virus might not have been ideal in this case either, although there are lots of rapid cell divisions in blood cells. You can imagine that a virus that can only gain access to a cell's nucleus when that cell itself is dividing would not necessarily be desirable as a vector of a transgene, especially if you were targeting other cell types that were non-dividing or really slow to divide. Then per injection event, per application of the therapy, you wouldn't get a lot of cells that could be targeted. So it's probably not as big of a problem with a blood disease as with some other diseases, but it was probably still, they didn't capture some cells that just happened not to be dividing when they applied the therapy. Another problem was that the reverse transcription, right? So if a foreign RNA enters the cell's cytosol, it can get rapidly marked for degradation, right? Because it doesn't have the poly A tail and the methyl G cap that it needs, and it's not a microRNA, so it can get targeted for degradation pretty easily. And if not, the viral vector itself can be targeted by the immune system before it even gets to the desired cells, which of course is not a big problem for people with SCID because they are severely immunocompromised. Okay, but more pressing and relevant to the rest of the lecture today is the fact that if you're using a viral vector that's inserting DNA into the host genome, a lot of the integrases that these retroviruses have insert quasi-randomly into the genome, right? So there's some evidence that the integrase targets insertion into gene-rich areas of the host cell's genome, probably because gene-rich areas tend to be highly expressed. They have a lot of enhancers and promoters in the region that express those proteins. So you can imagine if you're a virus, it's in your interest to land in a part of the genome that gets transcribed a lot, because then you're actually having your viral proteins being transcribed and translated, as opposed to living in sort of an expression desert of the genome, which might not be in a cell's interest, or in a virus's interest, excuse me. Okay. But if you're being inserted into a gene-rich region, there's also the risk that you're inserting into a gene, right? A really important, an essential gene even, right? So if you insert into an essential gene, you can kill the host cell, okay? Which is problematic for a virus, but maybe not as problematic as if you were to, not as problematic for the host, as if the DNA inserts into an oncogene, right? Or disrupts a tumor suppressor gene or something like that. So if it lands in the wrong gene or turns off the wrong gene because of inserting in the middle of the gene, then you can get cancer phenotypes, which is a huge problem, okay, as you probably learned when you guys talked about gene therapy. The other thing is that these viral vectors are usually constructed, or were historically in the 90s when people were dumb, designed with promoters and enhancers that drove transgene expression, so in this case the ADA gene, really to really high levels. Okay, so the viral vector itself that they constructed was expressing the ADA gene at really high levels. Okay, but what they found out later, which they didn't know at the time until the clinical trials were underway, was that that transgene, the promoters and enhancers used to drive the transgene, also transactivated oncogenes nearby, right? So not only could they insert into a gene, causing a mutation that might ultimately result in cancer or an increased probability of cancer, but you could also land amid genes and overexpress them, and that overexpression itself could trigger cancer as well. Okay, so these two are kind of like a double whammy of bad news for these vectors. So they're inserting in an off-target manner, and often they're inserting in regions that are already being highly expressed, and they're increasing the expression in those regions, all of which are bad. Okay. So this is the original paper that the French team published, and it was really exciting at the time. So this is 2001, so this is probably 10 years into the trial, into the clinical trial. And what they say in the abstract here is essentially that they used the Maloney-Murine leukemia virus, and they actually didn't target the ADA gene. They targeted a different gene, an I think interleukin-2 receptor or something like that. So it was another gene they knew a lot of people with SCID had a defective copy of. And they actually restored the function of the gene, and in many cases restored almost completely the immune function of the individuals, the patients in the clinical trial, indefinitely. Okay. However, of the 20 patients in that original clinical trial, I think 4 or 5 of them ended up getting leukemia, so a blood cancer. And when they looked at the mutations responsible for those blood cancers, they were always at the insertion sites of the transgene, of the functional AD, or in this case, I guess, interleukin-2 gene. So not only are they getting cancers, they know that those cancers were caused by gene therapy. So there was a huge controversy in the early 2000s because of these cancers, right? One of the 4 or 5 individuals actually died of leukemia. The other ones got chemotherapy and have since recovered from it. I believe, perhaps excepting one who died of something else, I can't remember, or died of some other complication. But the point is, there was a big debate in the media and in the scientific community as well about whether these gene therapy clinical trials should continue given the risk of cancer. Okay, so here is a review that I also provide to you guys in the additional resources section that talks about how this didn't come as a surprise to the clinical research team. They knew that this was a risk, and they informed the families that this was a risk associated with gene therapy. And the families essentially, with their kids who had SCID, their options were bone marrow transplants, which might not have been possible if there was no bone marrow match. And it also still, even to this day, has a really high mortality rate, right, 20 to 30%. So it's still a really risky surgery, big deal surgery. So there weren't many other options at the time. So these families consented to the gene therapy, knowing full well that was a possible outcome. Okay, and of the 20 patients in the clinical trial, all of them, I believe, with the possible exception of one. The details are here. I can't remember exactly, but it had a tremendously high success rate. Um, had now a functional copy of interleukin-2, and have essentially, though not completely, normal immune systems, and don't have to be taking all kinds of um, immunological supplements for the rest of their lives. So the argument that this writer is making is essentially that these clinical trials should continue because even though they're killing a few people, the disease is killing more of those people than the cure would have. And plus, they're learning about what the vectors are doing wrong and how they can improve and increase the survival rate and reduce the cancer rate as they go with the clinical trials. But as far as I know, these clinical trials were shut down. So they stopped the clinical trials after they had the evidence that the individuals were getting cancer because of the gene therapy. And I don't know the status of current gene therapy clinical trials in the US. Yeah, I'm not sure about that. But my guess is that they're probably the policy is probably going to shift very soon, given um, given the new hotness. Okay, so um, enter CRISPR-Cas9, right? So remember that some of the major problems with the old delivery system was that they were relying on the vector to insert genes quasi-randomly into the host genome, which could cure the patient or could give the patient cancer. So we didn't have any tools that helped us target particular genes or particular regions of the genome with extreme precision. Okay, so the good news in terms of what the status of gene therapy is these days is, first of all, that the viral vectors have generally been improved, although they do still present quite a non-trivial obstacle to gene therapy. So what they're doing now is those enhancers and promoters that used to just drive high expression of the transgene are now tailored a little bit more, so they're very specific to the transgene and don't transactivate a lot of other nearby oncogenes. So that's one thing that helps. And also they've selected better viral vectors that actually do a really good job binding to target host cells in vivo and infiltrating, shuttling into the nucleus without having to have cell division and things like that. But more relevant to us again is that off-target insertion is now much less of a problem thanks to technologies like CRISPR-Cas9. So CRISPR-Cas9 is one of several technologies now available that can insert genes or cause small indels, insertions or deletions, in a targeted genomic region. And they were actually discovered in the 80s when a bunch of researchers from disparate fields of microbiology, right? So they were discovered in lots of different species of bacteria, no one knowing what they were or that they all shared a really common function. Okay, but essentially what happens is that let me show you this and I'll go back to that slide. Okay. So CRISPR, by the way, stands for Clustered Regularly Interspaced Short Palindromic Repeats. Okay, so if you were to look at a CRISPR locus, like these early researchers looking at bacterial genomes were doing, you would see these regions being repeated, so the red, over and over and over again in this little clustered area of the genome. And inside those repeats, there would be little sequences that were palindromic, okay? So that were complementary essentially to each other if they were folded in half right there, okay? And then between those repeats, there were these interspaced regions, so called the spacer regions, and these did not have the same sequences from one to the other to the other. And in fact, years later, and actually decades later, um, they realized that these segments of DNA are actually viral DNA, right? So there's just these little chunks of viral DNA found in the bacterial genomes um, that are interspaced among these short palindromic repeat regions. Okay. Also, a couple of decades later, they started discovering these genes upstream and downstream of the CRISPR loci called Cas genes, so CRISPR-associated genes, that's what Cas stands for. And most bacteria have lots of different Cas genes, and there's not a lot of homology from one bacterial species to the next, even in closely related species of bacteria. So that's probably not a big surprise that it took people a long time to find Cas genes because there was not a lot of homology from one species to the next. But these Cas genes, they know, are always interacting with the CRISPR loci, but no one knew how or what they were doing for a very long time. Okay, so now we can go back to this slide, and I can tell you what they are actually doing. So what they were actually doing is these CRISPR loci were serving, I kid you not, as an adaptive immune system for bacteria, which is not a canonical thing that bacteria do, right? No one expected or thought that bacteria had any sort of way of having an adaptive immune system. And not only that, but this is immunity. The CRISPR-Cas system is immunity that bacteria can acquire and then pass on to their offspring, which is also a non-Mendelian inheritance pattern if you think about it. So it's fascinating from a transmission standpoint as well as a biotechnology standpoint. So anyway, these little regions of the repeats and the spacers essentially get processed, chopped up into little bits, and the spacers bind complementarily to a viral DNA or viral RNA that infiltrates the host cell. Okay, so if this host cell has CRISPR loci that have been exposed to this virus before, okay, then those CRISPR loci can bind to the new viral RNA that's being inserted into the host cell and degrade it. So essentially interfere with the viral replication cycle, okay, thereby rendering the bacterium immune to the viral infection. Okay. All right, so the player I haven't talked about yet is Cas9. So I mentioned in most species of bacteria, there are lots of different Cas genes, right? So there's a Cas1, Cas2, Cas3, et cetera, But there's one particular species, Streptococcus pyogenes, that has one Cas gene, Cas9, that is necessary and sufficient, i.e. causative, for cleaving DNA. So causing a double-stranded break of a target sequence, right? So in the case of the bacterial resistance to the virus, it would cleave the viral RNA that's being inserted into the host's genome by the virus at exactly the region, where the spacer complementary DNA binds to the virus. Okay. So this is a detail from a paper. It's a pretty complicated paper, so you don't need to really know what's going on here in specific detail, except to know that this little cartoon here in A is a schematic of Cas9. And Cas9 has several domains, right? So little regions of the protein that do different things. So they actually made two different mutant proteins. So they mutagenized the Cas9 protein in such a way that this protein domain was rendered non-functional in one of those mutant proteins, and this protein domain was rendered non-functional in one of the other mutants. Okay, so they have a wild-type Cas9, a D10A Cas9 mutant that's non-functional in this region, and an H840A mutant Cas9 that's non-functional in this region. And by non-functional, I mean they caused one amino acid substitution, either at 10 or at 840. Okay. In both cases, it was an alanine that was the substituted amino acid. When they did this, they looked at cleavage, so whether the DNA gets cut or not. Okay, so I can't go into a ton of detail about it here because I haven't really yet told you about CRISPR RNA or tracer RNA yet. But suffice it to say that the Cas9 has two protein domains that that are required to cut the two strands of the RNA target. Okay, so one of the protein domains is responsible for cutting the complementary strand. Okay, and that is this domain, right? Because when you use a mutant, it doesn't cut the complementary strand that was required to make the smaller molecule. And the D10 region cuts the non-complementary strand because when there's a mutant there, you don't get the molecule being cut at the non-complementary strand. Okay, so essentially what's happening, right? That figure does not have to make a lot of sense. I just wanted to show you there actually was some rigorous molecular work that went behind figuring out how all of this works. So what's actually happening is Cas9 is this big blobby protein in the background. And here's our target DNA or RNA in the cell, right? So maybe this is DNA that's been reverse transcribed by a virus, okay? And it's just hanging out in the cell. And Cas9 comes along, and one protein domain, right, this top bubble here, cuts the non-complementary strand, okay? And this other bubble, this protein domain, cuts the complementary strand. So Cas9 has two different domains, each of which cut one strand of DNA. So together, causing a double-stranded break, right? Cutting at both strands. So those are some, but not all of the components of the CRISPR-Cas9 complex. So here are a few more. So this is Dr. Doudna. If you watch the YouTube video from the schedule, you will know full well who she is. She is one of the two co-inventors of the CRISPR-Cas9 system here. So look out for her. She might get a Nobel Prize someday. But she's explaining essentially what's happening here. So we already know Cas9, right? That's this big chunky blue molecule, okay? And we already know that Cas9 is the enzyme that is responsible for the nucleolytic activity, right? So it's a nuclease, right? It's an endonuclease. It's cutting DNA, and it's cutting both the complementary and non-complementary strand. Okay, so it's creating a double-stranded break. That's what Cas9's job is. Okay, but there are also these two other molecules and this one other feature that I want to talk about. One of those molecules, this guy here, that looks like a toothbrush, okay, is the CRISPR RNA. Okay, so the CRISPR RNA has a sequence on the 5' end that is complementary to our target DNA. So in the case of the bacteria, this is the spacer region that has the viral DNA, and it would be binding to the virus's DNA or RNA here. Okay, this is the palindromic repetitive region, right? On the 3' end here. Okay, and the tracer RNA also has two parts here. One part is complementary to the palindromic region, okay, the repetitive region of the CRISPR RNA, and the other part of the tracer RNA forms this secondary structure, right? Forms this little hairpin, and they found out actually is responsible for recruiting Cas9 to this particular DNA sequence. Okay, so essentially, CRISPR RNA binds to the target DNA, tracer RNA binds to the CRISPR RNA, and recruits Cas9, and Cas9 is what makes the cut. Okay, the other important feature here is the PAM sequence. Okay, so the PAM sequence, they realized none of this will happen, none of these cuts will be made if there aren't two Gs, so two guanines in a row on the sequence just upstream of the CRISPR RNA region of complementarity. Okay, so this region is about 19 base pairs long. So you can find any 19 base pairs in a genome and engineer a CRISPR RNA that's complementary to that 19 base pair region. Okay, and it will create a double-stranded cut and exactly and only exactly that 19 base pair sequence in the genome, provided that there are two Gs immediately downstream of that 19 base pair region. Okay, so you should be familiar with Cas9, you should be familiar with CRISPR RNA, you should be familiar with tracer RNA, and you should be familiar with the PAM sequence. Okay. So what Dr. Doudna did is she and I think Martin Janek in her lab engineered just a little stretch of DNA, essentially, that fused the CRISPR RNA to the tracer RNA. So it became what's called a single guide RNA, or an sgRNA, right? So they just made a little loop here of DNA, and this all became one molecule of RNA. Sorry. Okay. So let's talk about why does any of this matter? Okay, we know Cas9 generates double-stranded breaks. Okay, so the Cas9 with the single guide RNA can generate a double-stranded break in DNA. Okay, why on earth would you want targeted double-stranded breaks? Why is that a good thing? Okay, well, it turns out that if you create a double-stranded break, most DNA repair mechanisms in most organisms will fix it. And they will fix it in a specific one or two specific ways. Okay? One way they'll fix it is they'll join those two broken regions back together. And that DNA repair mechanism is known as non-homologous end joining, or NHEJ. Okay? And when there's non-homologous end joining, it's not perfect. So often when those two regions are joined back together, they have small indels, right? Small insertions or deletions in them, which, hey, guess what? Is exactly what you want if you're trying to create a loss of function allele at that site, right? So if you want targeted mutagenesis, then that's what you would do. You would create a double-stranded break and then just let the cell do what it does naturally, which is non-homologous end joining, if you want a knockout mutation at that site. Okay. If you want a knock-in mutation, then you'll have to rely on a different cell DNA repair mechanism called homology-directed repair, or I'm guessing the HR here stands for homologous recombination. So essentially what, in this case, you would have to do is create the double-stranded break and then provide some kind of template DNA that has regions of homology flanking where you created the double-stranded break. So it has to have regions of homology on this end and on this end, okay? And then the cell will find that region of homology and essentially recombine that donor DNA into where the double-stranded break was to repair the double-stranded break, okay? So that's what you rely on if you want to make a knock-in mutation, right? So if we wanted to insert a gain-of-function allele into the ADA locus or the interleukin-2 locus, we would have to provide donor DNA that had the gain-of-function copy. Okay? So that's the difference here. If you don't include any DNA along with your your Cas9 single-guide RNA, then you'll generate a knockout, okay? But if you include the donor DNA to get homologously recombined in, you'll create a knock-in. That's how you can insert new genes. Okay, so Cas9 and single-guide RNA, so CRISPR-Cas9, CRISPR-Cas9 complex, can do this, can create these targeted double-stranded breaks. But they are not the only technology that can do that. We also have zinc finger nucleases, or ZFNs, or TALENs, whose acronym escapes me now, but I think it's like transcription effector like something nucleases. Forgive me. But with either of those, essentially what's happening is, well, all three of them are, you could think of them as designer endonucleases, right? Where a researcher can engineer exactly what sequence of DNA they are targeting and making a double-stranded break at and cutting. Okay, so they're all designer endonucleases. Okay, so you know restriction enzymes like EcoR1 or BAMH1, which recognize and cut a very specific sequence of DNA. Okay, well, the problem with something like EcoR1 is it recognizes I think it's a 6 base pair sequence, right? And that particular 6 base pair sequence, just by probability, will occur many, many times in a genome, which is why when you digest a genome with EcoR1, you get lots of small DNA molecules, right? So it just cuts up, it just chops up everywhere, right? But if you have a 19 base pair long sequence that gets recognized and cut, probability-wise, that's going to happen exceedingly rarely, and usually, you know, if you're lucky with your design, only once in a genome. So you want these designer endonucleases. They are a desirable tool to have in our genetic toolkit because they target a very specific sequence of DNA that is unlikely to occur many times in the genome. Okay, the difference between CRISPR-Cas9 and what TALENs and zinc finger nucleases do is that CRISPR-Cas9 uses an RNA complementarity to your target DNA to cut. Zinc finger nucleases and TALENs rely on proteins binding to your target DNA to cut. And guess what? Generating certain proteins that bind to DNA in a certain way is a much tougher engineering feat than generating an RNA with complementarity to your target sequence. Okay? So zinc finger nucleases and TALENs are a lot tougher to design than CRISPR-Cas9. Okay, so that's disadvantage 1. To zinc finger nucleases and TALENs compared to CRISPR-Cas9. that doesn't mean that they haven't gained popularity, right? So you can actually go online and buy a kit, right? A talon and so the E was an effector. It's an effector nucleus. Um, you can buy kits, talon kits, for about $425 online. But you could buy a CRISPR-Cas9 vector for $65, right? So CRISPR-Cas9, in addition to being much easier to think about, to engineer and design, much easier to implement, it's also an order of magnitude cheaper. Okay, so this is why everybody is freaking out about CRISPR-Cas9, is because it is making transgenics extremely affordable. Okay, so that's it for CRISPR-Cas9. Some credits and additional resources. A lot of the images that I showed you today were actually from Dr. Doudna at Berkeley. So I borrowed a lot of her images from her YouTube video. And I'm also linking you guys to a couple of papers that I read that were really interesting about gene therapy and about CRISPR-Cas9, all of which actually I have linked to as a zip file if you follow this link. All right, thanks a lot for your time, and I hope that you have liked this course. Take care.